look at the top, they start with the intruder. And that can trigger your innate defenses, right, that we talked about, like complement or interferons. But it also can trigger the adaptive defenses, the specific defenses. The B cells, which, if you have the right clone, the membrane-bound antibodies can bind the free antigen. But now we're going to talk about the T-lips. And the big difference is that the T cells can't bind the free AGs. They need an antigen-presenting cell to present a piece of that antigen to it to cause activation. They still require exposure, but they require APCs, stands for antigen, presenting cells. There are not too many cells that have this function. The antigen presenting cells include B lymphs, they include macrophages, or dendritic cells. They, okay, that's it. They have this function of presenting pieces of non-self antigens to activate the T-lips. Now, if you look at this figure, there's crosstalk between B and T. The crosstalk is the helper gene. They get the whole thing going. Okay? They can activate the B cells, so that they're extremely important to the whole process. That's the other thing we need. So we need to know about the APCs. We need to know about this cell-mediated immunity. Okay, not antibody-mediated, cell-mediated, the T cells. And so there's a concept for T cells called MHC restriction, because there's the CD4s and the CD8s, these different subpopulations of T lymphs. MHC restriction. Well, what it is shown here. You have lymphocytes, T cells, that proceed from the bone marrow to the thymus. B cells don't do that. T cells do. T is for thymus. And they go through those two rounds of selection. We gave the example of the CD8s interacting, around a positive selection. You can interact with MHC. The second round is a round of negative selection. You don't react to the self-antigen. Those are selected. And what you see here is the CD4s, they're restricted to interact with the MHC class 2 on those APCs. The CD4 cells, they become T helper cells. They're restricted to interact with MHC class 2. Then CD8 cells, those become the cytotoxic T cells. They're restricted to interact with MHC class 1. So that's this figure is showing you, basically. Once they interact and are activated by the MHC class 1 or 2, they become the cytotoxic T cells or the helper T cells here. So here's this picture in outline form. Just remember, MHC restriction requires activation, but there's the restriction is class 1 is restricted to the CD8 subpopulation. Class 2 is restricted to the CD4 population. So let's kind of get into detail what you should be thinking about for the different classes, because there are differences, obviously. There are different classes. Class 1, Class 2. We'll start with Class 1. I think it's a good idea to keep these 3 questions in mind for each class. Class 1 MHC. They're recognized by cytotoxic CD8 cells. First question. Which cells present Class 1 MHC? Okay, this is the one that's widespread. All cells that have a nucleus will present the Class 1 MHC. Okay, now most cells have a nucleus, so this is no cell. Maybe the exception is red blood cells. They don't have a nucleus. All cells with a nuke present this MHC class 1. Okay, I'll say not RBCs. Any cell without a nuke can't do it. They don't have the genes and proteins, obviously, because they spit out the nucleus. Okay, that covers most everything in the body. Okay, so good. They have the class 1. Now, what do they display in the groove of class 1 MHC? They display an endogenous antigen. Endogenous AG. That means from within, generated from within. This is a piece of fragment from within your own cells. And it's displaying it on MHC1 so you don't get attacked by your own immune system. Okay? Now, so when you're not sick, it's going to display, and that answers the third question. What kind of antigen is it? Is it self or non-self? Well, if you're not sick, you don't have any infected cells. It's going to be a self-AG. So you can display an endogenous AG. It could be a self-AG, so that way when your CD8 cells come on by, they don't trigger a response. A response is not triggered in the CD8 cells. If you are sick and you have some virus-infected cells, those cells will present a non-self AG. And that's where you want the T cells to respond. Well, here's a picture of that. Here is the endogenous antigen, whatever it is. You're degrading it and you're putting a piece of that in the groove of MHC1 and you display it externally so that when immune cells go by, the CD8 cells, they'll interact with class 1 MHC, but they won't be triggered by a self-AG. Okay. Of course, if it is non-self, it would be, but just keep in mind, is it self or is it non-self? Now, class 2 is different. Class 2, we'll ask the same questions. Which cells present class 2 MHC? Only the ones that have the APC function. Only cells with that APC function role, if you will, present to MHC class 2. And I already listed the 3 kinds of cells: dendritic macrophages and B cells. So what do they display? They're displaying, by design, an exogenous antigen, something they've eaten. So in all cases, it's exogenous because these white blood cells, for example, macrophages, they're always going to eat non-self, always. That's why they've phagocytized it in the first place. It's an exogenous antigen that they've eaten. So when they display a piece of this on their groove in the MHC2, it's always non-self. always the result of an infection you're mounting a response to, and that's shown here. So what you see here is, I don't know, let's say it's a macrophage, and you engulf this pathogen, this bacteria, as they call it. Well, it's already been phagocytized. I mean, this is already a recognition of non-self. Okay, so you've already destroyed it, but now you want to mobilize the specific defenses against it. So what you do is you put a groove of it, you put a piece of it in the groove of your class 2 right there. And so this should trigger a response in the CD4 cells. And so it all begins with the CD4 cells. Let's talk about the T cell activation of the CD4 cells in 3 steps. The CD4 cell needs a TCR, T cell receptor, to recognize MHC2, which we already know it does. But the TCR, it's got to be specific for this particular non-self bug. If that's correct, you also need a co-stimulation to complete the cellular activation of CD4. And you also need interleukin 1. So that's a lot of things to keep track of. Well, it's only 3 things. But well, here's a picture of that outline. I think it helps to draw it out. So what we're doing here is, okay, let's say we're sick. We're mounting a response. We first recognize it, and we activate the CD4s. They're the helper cells. They're going to help everything else get activated, so it's good to start the discussion with their activation. Activation of a CD4 cell. So what they show here on the left, that's the APC. It's presenting MHC class 2. I'll just put a Roman numeral 2 for MHC class 2. And it's presenting a piece of the phagocytized antigen. Say it's a virus. We'll call this viral AG, non-self, right? In red, viral AG. So what happens is the T cell comes along, CD4 cell, and it has a T cell receptor that's specific for this viral AG. So number 1, it interacts with MHC class 2, and it's specific for this viral AG. So I'm going to call this receptor TCR, T cell receptor. I don't think that's hard to remember. Now what stabilizes this is the CD4 surface molecule in that groove of MHC class 2, just like they have there. So this is CD4, the actual CD4 molecule that they named the cell after. And this triggers intracellular signal, call it signal 1. But it's not enough. You need a second signal. So that's why you have this co-stimulatory process. You have other ligands, like for example, CD28. The APC may present a ligand called B7. That fits it perfectly. And they call this co-stimulation, the B7-CD28. Co-stimulation. Well, that gets a second signal, And that completes the activation along with IL-1. We'll call this signal 2 the B7-CD28. Well, anyways, both of those should complete activation. Let me not forget IL-1. IL stands for interleukin, and that's easy to remember. This is a molecule secreted between leukocytes, interleukin. It's helping to complete the activation. IL-1 will help complete the activation of this CD4 cell. So signal 1, signal 2, IL-1 completes activation. T cell activation. And an activated T cell has different properties. An activated helper T cell, now activated, it will start displaying different cell differentiation markers, like on this slide, CD154, shown here. This shows signal 1, signal 2, right, from both interactions. So it shows other things like CD154, which upregulates inflammation. And an activated T cell will in turn secrete IL-2 to activate other things. So it'll display CD154. So basically, increase inflammation. Secrete IL-2. Before proceeding, let's take an inventory of our outline here. I think you should have drawn it out with me. Number 1 says TCR recognizes MHC2, so these two receptors there. Number 2 says this TCR is specific for this viral AG, the red ball. And I said CD4 stabilizes this. I drew CD4. Number 3, the co-stimulation. That's the B7-CD28, which I have on the outline. And I also said IL-1, secretion from the APC to the helper T cells. All of those things are going on. So now we have an activated helper T. Any questions on this picture? We're going to see what the helper T does in turn. We're going to see how it helps. Well, if the CD4s don't work, it may be because the co-stimulation failed. So if you read what I defined for anergy, without co-stimulation, anergy occurs, which pretty much means you don't get a response. And without a response to the T helper cells, there is no response overall. So that's why this whole process is very important to understand. Well, let's say it works, and you have the helper T cells that are now activated. What are they doing? This shows a couple of things. How do T helper cells help? I'll put in the form of a question. What you see here is it's binding to a dendritic cell. The T helper cell, it's helping the dendritic cell to express those co-stimulatory molecules like B7. T helper cell helps dendritic cells express co-stimulatory molecules. B7 is an example that I gave on the previous slide. Also, it's helping The T helper cell is helping the CD8 cell with the help of IL-2. So the T helper helps the CD8 cell by secreting IL-2 to complete its activation. So IL-2 is completing the activation of that CD8 cell. It also helps the B cells. Here we see a helper T cell. It's binding the complex of the B cell. That's displaying the MHC2, right? We said it could do that. It has the APC function. And IL-4 from the T helper cell completes the B cell activation. That says helps. So the T helper cell helps complete B cell activation with IL-4. There's a lot of interleukins. I've just given you 3 to know. IL-1, IL-2, and IL-4. So we already know how the activated B cells are supposed to work. They're supposed to proliferate, increase the antibody titer, they become plasma cells, they have memory cells. But I didn't mention much about the CD8 cells yet. They perform hand-to-hand combat. They target virus-infected cells where they administer a lethal hit, shown here. So activated cytotoxic T cell. They target virus-infected cells and administer a lethal hit. So when we say lethal hit, what we really mean is pay attention to the perforins and granzymes on this figure. The perforins perforate, they punch holes. That allows the granzymes to enter. And the granzymes, they kill the cell. They are proteases that degrade cellular contents, and they stimulate the apoptosis of the target cell, the virus-infected cell, the cell you're trying to destroy. Okay. And that's it. That's all I have for immunity. Let's remember that the multiple choice portion of the test will become available sometime today, maybe tomorrow. Today or tomorrow. It'll be available. When does it due? Sunday. But right now we're going to take the written portion of the test. So I'm not going to reseat you or anything. Just clear your desk. Get something to write with. Get a divider. Once your desk is clear and you're properly divided, I will give you a test. Right now.